This is a special episode of the Stem Cell Podcast, ISSCR Day 4 with Dr. Sheila Chari. Hey everybody, we're Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. This week, Arun and I have been attending the 2021 ISSCR annual meeting and releasing special episodes discussing some of the highlights. We've also been chatting with special guests, including Dr. Sheila Chari, Editor-in-Chief at Cell STEMCELL, who we'll be hearing from in just a bit. What's more, we've taken the plunge to release these episodes not only in their traditional podcast format, but also as video episodes, which you can find on the Stem Cell Podcast website or Stem Cell Technologies YouTube channel. We're going to kick things off in just a minute, but before we get to that,
check out tomorrow morning's plenary session on cell therapy and tissue engineering for cutting-edge approaches by Sonia Schrepfer, Shubing Chen, Paul Tessar, and Wolfram Zimmerman. The session is sponsored by Stem Cell Technologies, which provides reagents and supports for HPSC-derived cell and gene therapies. For additional information on how stem cell can support your research from discovery to the clinic, visit the Stem Cell virtual booth. And we'll pick things up from where we left off yesterday, and that is at the ISSCR Achievement Award session, headlined by none other than Dr. Janet Rossen. From embryos to blastocysts and stem cell embryo models, she's talking about her journey in understanding why development matters, and in particular, talking about early embryo development and the critical regulators of that process. So, for example, she talked about how the inner cell mass and trophectoderm lineages are established, and the role of hippo-YAP signaling and in controlling early lineage establishment in the blastocyst. And I think the future-facing direction of the talk was the next steps. So, can we capture totipotency in a dish? I think this is a very, very hot topic, and perhaps the biggest unanswered question in this particular subfield of stem cell biology is really being able to capture totipotency. We're not quite there yet, as she alluded to, but some of these early embryo models that we've discussed on the show, like blastoids, gastroloids, all that, they're going to help us unlock some of these key mechanisms, which may ultimately lead us to that dream of achieving totipotency. And the other reason why I like these award sessions and these achievement awards in particular, it allows for these esteemed dignitaries in our field to really give a big, 10,000 foot view recap of their time in stem cell biology, which I think is really informative for the trainees, and particularly the younger trainees in the audience. Because for those of us who have been in the field for a while, we know about Dr. Janet Rossen and other dignitaries in the field like Stu Orkin. But for the new trainees, this is all new and exciting and really critically important to their career development. So I think that's part of the reason why I really enjoy these talks.
Yeah, she was one of my favorite guests that we had on the show. So check out that episode. And like you said, she's able to see the whole organism, right? And that was kind of the theme of her talk, or at least the title was the whole view. It must be a real satisfaction for her in her career to go from the embryo. I mean, that was the title, to go from embryo to stem cells to stem cell based embryo models. And so it's kind of this full circle idea. And it's nice also to see her sharing the stage virtually, at least with Nicholas Rivron, who's going to be on the show actually in tomorrow's episode, because it's a nice counterpoint. I mean, he's carrying the torch, so to speak, and bringing these blastoid models into the next generation, along with many others. And just a little kernel that I thought that she ended with, which was nice for me, was the tidbit to show that she's still really driving the science forward and showing that there was a real not stark, but subtle, but real difference between the blastoids and the iBlastoids. And the recent story, they came out on the same day in the same Advanced Online in Nature, I think it was. And deconstructing with the lineage analysis and RNA-seq there, she showed that Jun Wu's blastoids were actually generating anion, whereas the iBlastoids weren't. So nuance there, where you can see that she's still really scratching at it and trying to get to the truth. So that was a really nice, real satisfying experience for me to see her talk. Like you, I really enjoyed that. And getting into the afternoon concurrence for me, the big win was Katsuhiko Hayashi. You know, I'm in reproductive biology is my major focus right now, and he's one of the guys early on. Came out of Mitsunori Saito's lab, and their group there, they were able to generate germ cells and ultimately live-born pups from ES-derived sperm, eggs. Hayashi focused on the oocyte, and then also showed that they could do it in human. But the real roadblock there, and this is conceptually big picture for the ISSCR, was the takeaway for me here is that, you know, we're building our own tools now biologically. The roadblock used to be that in order to get the primordial germ cells to form more mature you know, their derivatives, oocytes or sperm, you had to induce with you know, somatic tissue from the gonad, okay? In this case, from the ovary, somatic cells from the ovary, and they would actually, in the human system, take the cells from a mouse fetal ovary and co-culture them. And you can imagine ethically that's never going to fly in the human system. But what do they do here in the mouse system, of course, but this is really soon to come, I'm sure, the human, is they make those somatic cells from ES cells. So I think we're reaching an era now where if you need an inductive tissue, rather than trying to reproduce the milieu with the molecules and recombinant proteins and small molecules, et cetera, we're making the tissue. So we're really approaching a very, very high degree of recapitulating the biology of embryogenesis and organogenesis. So really exciting, I think, chapter for all of us as stem cell biologists.
Yeah, absolutely. This is a very, very hot sub area of our field in stem cell biology. And kind of building on that, I decided to go out of my comfort zone a little bit and focus on a theme session and attend a theme session titled Comparative Early Development. We're actually talking about some of these new embryo modeling technologies that Dr. Ross and you just talked about just now. So just wanted to recap a few of the trainees who are presenting some amazing work in this subfield. Hongmei Wang, who's deciphering and comparing the mechanisms of primate development and really trying to figure out the differences between human and cynomolgus monkey developmental processes using some of these models that we're talking about. Cody Keim, who has been in Japan for a little while, he's an American scientist, but he's based at the Riken in Japan, I believe. Synthetic embryology and talking about reprogramming epiblast stem cells into pre-implantation blastocyst-like cells. And he's also on the job market and just published a PNAS paper on that exact topic. Ivan Bezov, who gave actually a really neat in vitro model of the maternal blood vessel network, and basically modeling that using 3D bioprinting and organ chip. I had no idea that you could actually kind of recapitulate the maternal placental blood vessel network in that way. It's not often that you hear about tissue engineering in the context of studying like implantation. I thought it was really nifty. Chen Dong, who's looking at a genome-wide CRISPR knockout screen to identify critical regulators of the growth in trophoblast stem cells. Catherine Polkoff from NC State. Daylon and I have both lived in North Carolina for a little while, so we know a little bit about NC State, and in particular, how they're actually really well known for their large animal research. They have amazing large animal research facilities over there. So she actually had a transgenic pig model, CRISPR engineered pig that you can develop in grad school these days, apparently at NC State. So that's pretty neat. So she's using this pig model to study the divergence in the hair follicle stem cells between different species. So why, how are these developmental processes different in the pig versus other non-pig species? And finally, Sarah Bizzotto talking about the landmarks of human embryonic development that are inscribed in somatic mutations. So I think a really solid subsession, a really solid section of the conference focusing on early embryonic development and some of the amazing technologies that are helping us better understand it.
Yeah, I'm loving how they're clustering this. I mean, whoever designed this deserves a lot of credit because, it seems like, oh, accidental, like, oh, that's interesting. I'm interested in that too. And it's hard to even get out of the session. It's all by design, people. This is a really good management of the program, in particular virtually, because there's so many more talks you can see in this format, it seems, although you kind of burn out on it. My eyes are watering by the end of the day. But that brings us to today, this morning. This is day 5. We covered the morning as well. Some of the trainee sessions I like to highlight. There was Ryan Goes from UCSD. I liked this talk because it was a hack where you can modulate the enhancer region of FOXA2 in order to augment differentiation of pancreatic beta cells. And, in a conference where it's really coming to light the strength and power of base editing and CRISPR, you can envision a future where we're going outside of the normal ways of inducing cell types, with whatever cocktails, and instead engineering lines that are well suited to differentiation for off-the-shelf approaches. It's nice to see young trainees doing the work because you can see that their imaginations are totally unleashed and they see this world as a prerequisite to therapeutic application, not as some far out tech. Then also there was Talia Dayton. I just have to throw her in, Merit Award winner from the Hubrecht, because of course, hailing from the lab of Hans Clevers. She was working on pulmonary neuroendocrine cells, generating organoids from those in both healthy state and disease. And it really drove home to me something that Dr. Clever said in his talk early on, which is that these grad students and postdocs and trainees, they just come to him with these fully formed projects, and he pretty much just has to rubber stamp them. And when you see his trainees give their talks, you realize that my man is not exaggerating, it seems. I mean, he's probably very generous in his assessment. But also, I would like to have access to those postdocs because they really do just come with just a watertight story right out of the gate. Here, in this case, you could see that they're running through the entire list of patient-derived organoids. So that's really exciting. There were a lot of other talks I'm not going to get to for the interest of time, but I did try to catch this exciting talk. I thought it was going to be great about patient-derived organoids, in this case, glioblastoma organoids, but the tech broke and I couldn't hear the speaker. I don't think I was alone. There was a lot of O faces there. I mean, it's a bit of an issue with this conference. Every day there's a handful of these technical debacles. So I really can't wait to get back in person. Instead, though, I was happy to catch the other concurrent talk from Dr. Torres Padilla, who's from Helmholtz. And she was talking about, circling back to Rassant, about can we induce pluripotency in these 2C-like cells, doing a really rigorous molecular characterization of those. So that was really exciting to me, and that'll kick us into the plenary. For me, I mean, tech-wise, I didn't even know. I caught the first talk, caught them all, but I was really interested in the plenary and Amanda Fisher's talk just from the MRC. She was talking, I didn't know you could do this, but talking about sorting mitotic chromosomes and being able to distinguish each one individually or in clusters, and then doing some proteomics to see what's bound. And you can envision there's a whole other, forget about peeling back the onion. There's a whole scallion, at least, of science there where you can just use this kind of mitotic flow to unpack gene regulation. So that was really exciting for me. What do you think of the plenaries, Arun?
Yeah, I thought the plenaries were great. It was fun listening to Amanda Fisher and her adventures in mitosis is what I like to call it. Yeah. Again, I agree with you. I had no idea you could do that, purified native mitotic chromosomes from different species. That's pretty wild. I was really excited about Samantha Morris's talk. She's a relatively young PI who is an expert in all things single cell. And she's using single cell to dissect reprogramming and development too. She's got this new cell oracle tool, which is a way you can predict the regulators of cell identity in silico. It's really nifty. It's like a computational tool that can combine single cell transcriptome and ATAC-seq epigenome profile to actually infer the gene regulatory networks that are critical to early development and reprogramming too. So I thought it was a great plenary. And the next session that I wanted to highlight was, honestly, it's always one of my favorites every single year. This is the Women in Science session at the ISSCR. It's a session that was chaired by some of the most amazing scientists in stem cell biology. We had Janet Rossa, we had Chi Yoon, who's actually from industry, Dr. Christine Mummery, the president of the ISSCR, Madeline Lancaster, our friend and rising star in cortical organoid biology, and Valentina Greco. I mean, how that's just a superstar list right there of women scientists, women stem cell biologists who are just leading our field. It's we're, and a number of them we've been also very fortunate to have on the podcast. So we feel really fortunate in that way. They were talking about the unique barriers that are experienced by women in science and in stem cell biology in particular, and ways that we as a community can perhaps alleviate some of these barriers. The thing I really liked about this session, and even though I'm not a woman in science, this particular session was geared towards women and trainees who are looking to make the jump to independence from being a trainee to having an independent lab, which is kind of the position that I'm in actually right now. So there were, in my opinion, there were three major unifying themes of the session: the importance of mentorship, visibility, and encouragement also. And each one of the speakers was giving some really amazing stories as to how they got to where they are by a combination of extremely hard work and having people who helped lift them up. So in terms of mentorship, it's important to know from the perspective of a mentor and from the perspective of a PI, when to let go and when to let a trainee stretch their wings and really achieve that independence. It's tough. I'm not a PI, but I can imagine it's tough for a PI to actually let somebody go who's so exceptionally talented and also give that trainee a portion of the science being conducted in the lab. In the context of women in science, I think visibility is so critically important. Having women in positions of power, like Dr. Mummery, for example. And I think we're lucky in the stem cell field to actually have a number of women in positions of power in our field, and positions of leadership and accomplishment, like many of the panelists on this particular session. And encouragement. There was one particular story that Madeline Lancaster told where right before her big cortical organoid paper came out and was about to make a big splash, she was a little bit uncertain, a little bit lacking in confidence as to whether this would be a well-received story. And there was a female scientist from a really top institution that came to her and told her that she was doing just amazing work. And that was something that was able to really boost her confidence and, bring her on her way. So it's really critical to have that confidence. And hopefully this show is going to continue to serve as an avenue where that we can lift the voices of women in our field and be a positive avenue for change. So I'm hoping that this podcast has been a plus in that way.
We can hope. And I would advise all of you, like Arun, I'm sure you weren't the only Y chromosome in that session, but I would advise all of you who didn't tune in for that to catch it on the rebroadcast, because it's not just about being aware of that position, but also there's a lot to be learned, as Arun alluded to there. A lot of the things they're talking about in that session can apply to any trainee or anyone in science. So check it out, and you'll be able to catch Valentina again later today. She's giving the Momentum Award lecture at the meeting this year, also much deserved. And we're not going to be able to talk about that on this roundup, but we will talk about it tomorrow morning. Next, though, on to another woman in science in a particularly powerful role, editor-in-chief. We're going to hear the perspective of Dr. Sheila Chari. But before we get to that, I have a brief message from Stem Cell Technologies. Reduce risk and obtain greater numbers of higher quality cells in your human pluripotent stem cell-derived cell therapy development with Teaser AOF by Stem Cell Technologies, manufactured under relevant CGMP-S. Use Teaser AOF to consistently culture viral, safe, high-quality PSCs on a schedule that works for you with whatever cell lines you choose. Learn more at www.stemcell.com/teaser-AOF. All right, everyone, we're here with Dr. Sheila Chari, who is editor-in-chief of Cell STEMCELL. It's a Cell Press journal. Her primary responsibilities are knowing and publishing the top stem cell discoveries, driving journal publishing strategy, and managing a global editorial staff. She travels to international scientific conferences, when they're in person at least, and research institutions to be on top of the latest developments and meet with authors, reviewers, and readers. She's a proud member of the stem cell community and an ardent supporter of stem cell research. Sheila, thanks so much for joining us today. Tell us first, what have been your kind of highlights so far for the conference? How do you think it's going?
Well, first of all, thanks so much for having me back on the podcast, Daylon and Arun. It's great to be here with you. I've really enjoyed the meeting this week. I've been surprised by how engaging the format has been. I thought, oh boy, this is gonna be a whole 2 days longer than it typically is. How am I going to keep up? But I've enjoyed the way that the sessions have been thematically organized. So, what are my highlights? So, probably my favorite talk was Stuart Orkin's award lecture. I really loved how he talked, the full span of how he went from a basic biological discovery on how enhancers operate all the way to a clinical outcome for sickle cell patients, and seeing the different strategies that are being used was really fantastic. When those two papers came out in December of 2020, it was pretty dark days in the pandemic, and I don't think I really was able to take time and stop and really look at that achievement and what we can really do for patients now. And the approach that they've used is really kind of counterintuitive. It's not just gene replacement, it's using basic chromatin mechanisms to help reactivate a fetal gene. So I thought that was just a fantastic talk. I would also say that the theme of modeling early human development has come across really strongly at this conference, and I've enjoyed it so much. The first panel that Janet Rassant did on, I love the way they titled it, Fascination with Gastrulation. I wish I had thought of that. Was fantastic for kind of setting the stage. And then you saw over the course of the conference so far, people touching back on that, from Matthias Lutoff talking about modeling heart development, gastroloids, to Janet's own talk, where I really appreciated the way that she took that comparison of what we know about mouse early development from embryology and the stem cell models, and then compare that to where the human systems are right now. I thought that was really fascinating. And to see Nicolas Rivron talk about his work modeling implantation, I thought that was pretty cool with the co-cultures of the endometrial organoids and the hormone stimulation. Those were some of the moments where I was just so enthralled by the talks.
Yeah, absolutely. I think Daylon and I were both talking about Dr. Raron's talk and how that was like one of our favorites. The fact that you can model that process of implantation ex vivo, in vitro, using these endometrial organoids, that's just still so mind-blowing to me. I wanted to get your perspective though. So, you are the editor-in-chief of Cell Stem Cell. So I wanted to get your perspective as an editor as to like what you prioritize. There's so much that you can attend in a conference, right? And now that we're all virtual, it's easy to bounce back and forth between different sessions and pick and choose the talks that you want to attend. But as an editor, are there particular strategies that you take towards prioritizing certain sessions and talks to attend?
Yes, there is. And, this year is different than how I typically approach ISSCR. So for me, on a normal year, ISSCR is all about networking. It's the only moment where everyone from all over the world is going to be in the same place. And I use that as an opportunity to meet with as many people as I can. And the talks are kind of the icing on the cake. Obviously, this year it's a little different, so I had to approach it differently. So what I looked for was the themes that seem to be popping up over and over again that are most exciting to me as far as talks to prioritize. And then here again, the early modeling, early human development is just blowing up. And so every talk that focused on that was something that I made sure to go to. But I also thought about how can I replace the networking that I would normally do. And I think ISSCR has done a fantastic job of having kind of smaller sessions that are on topics that are adjacent to the science itself, that I made a priority to go to and felt like I got a lot out of. So for example, the of the policy and science communication panel. I thought that was fantastic. At STEMCELL, we really tried to also cover those issues. And with the new ISSCR guidelines being issued, I want to stay on top of that. I want to know who's really engaged in this process, what issues are coming up down the pike that we should be aware of, so that we can be looking to cover that as well for our readers. Because a point that I think Robin Lovell Badge made during one of those talks was that, this is our responsibility as scientists in the community to be informing the public and policymakers about where the science is and what the implications of that are for policy. And so if we don't engage in that, someone else will. So those are the types of things that I do also prioritize. So I went to the early career researcher discussion as well. I thought that was fantastic. It's something that's shifted for us at the journal over the past year. I think once we realized we couldn't meet people in person, we really had to figure out how to continue to support our mission, which is to be like the journal for the community and to support everyone and bring them together. And so hearing firsthand from researchers about what they've experienced and what concrete things actually help them was useful for me. And it helped me think about what I can bring back to my publisher and talk about in terms of supporting people during this really important stage of their career.
Yeah, I noted in the past couple of years, I don't know, maybe longer, that the journal itself, Cell Stem Cell, or maybe it's the wider press journals as well, but I obviously have more of a focus on Cell Stem Cell. There's a lot of things that have been added. It's evolved, like everything. A lot of journals evolve in terms of the content. I know there was like from the mentor, there's like a thing where the mentors talk to more trainees, focus, and it helps to, I think, for your younger readership, because at the end of the day, the journal exists for the forum, right? It exists for the scientific community in this case, or whatever you want to call it, the readers. And I don't want to boil it down to you're trying to sell journals, right? But like at the end of the day, you're trying to get the science in your journal consumed by the widest scope of scientists and to have the greatest influence and for the betterment of mankind and all kinds of altruistic goals. But at the end of the day, you do have to kind of shape in your position the mission and the format and the scope of the journal. Is that something that you like the guidelines this year, they circle back on here 5 years on and there's a revision? How often do you find that you're circling back with your peers at the Cell Stem Cell or Cell Press journals largely and talking about the format, the nitty gritty, the way the journal is presented, and specifically like in terms of the science here? Like you come to these conferences and is it like, okay, wow, well, here's the direction that we're going. In this case, you'd be like, wow, everything's human now. Organoids, we're past organoids, we're assembloids, we're vascularized. Does that kind of shape what you would expect to see in the next year from Cell Stem Cell, the kind of things that are really at the forefront, not just because they're new now, but also the direction of the science? Do you have to constantly revise that?
Yeah, for sure we do. And I would say we do it throughout the year, but we also take moments to step back and really look at the field and share our insights on what's moving and what we should move away from and what we should move towards, right? So probably once a year we do kind of our little like strategy retreat where we talk about our, what we've observed through conversations with scientists, through papers that we've really enjoyed. Throughout the year, we really survey the related literature. And so every other week, we have our own little journal watch amongst our editors and other people at Cell Press that are interested in stem cells and regenerative medicine. And so we kind of use that as a starting point and go back to it and really reflect on the trends that we see and what we want to pursue. And, how do we translate that? Well, we do things like we organize conferences that are based on issues that we think are really important. You both, thank you so much, participated in our engineering organ and organoids cell symposium about 2 months ago. And so that's obviously an area where we see a lot of growth. And so we want to bring together communities that wouldn't normally talk to each other so we can kind of be there at the outset and see how things shift over the next years.
So Sheila, I wanted to talk about kind of the current moment that we are in right now, this current moment in time. And the reason we're actually talking to you through a computer screen as opposed to in person is because of what's happened in the last 18 months, right? This global pandemic. And it's impacted everything, right? It's impacted science, it's slowed things down a lot of times, but in some ways it's changed, in my opinion, the publication process, maybe for the better, right? So, the review process was kind of expedited a little bit during the onslaught of COVID related papers that came out over the last year. And the grant review process was even accelerated a lot for certain COVID-19 centric grants. So, and of course, preprints have dominated the preprint servers like bioRxiv. And of course, CellPress has their own sneak peek and preprint servers as well. I just wanted to get your insights and your opinion on how the pandemic has changed science for the better. Acceleration in science is usually a good thing, right?
Yeah, I think we can look to some positive takeaways from what's happened over the past year. And I think we have to give credit to the stem cell community and to ISSCR for the way that they mobilized research about COVID-19. So when ISSCR first started organizing those virtual sessions, I could immediately see how collaborative and open people were being about sharing their data, sharing what they're learning in real time, and actively trying to forge collaborations and move forward very quickly because of the urgency of the situation. And so seeing that openness was really great for us, because then we could also expedite review. We could actually require that all COVID content be put on a preprint before we even published it, because it was important to get it out as quickly as possible. About a year ago, we instituted a new section in all of our papers called the limitations of study. And if you're interested in learning more about this, I encourage you to look at an editorial that we wrote about a year ago about how the changes that we're going to make based on COVID-19. And one of them was this limitations of study section, because we felt that an important part of the review process that was at the time happening behind the scenes was the discussion between editors, authors, and reviewers about what's enough for this paper, and what are the open issues that cannot be addressed in this one manuscript, and what's next, and let's talk about other interpretations without holding the paper back. Let's be open so that everyone can consider and discuss these things, and then take them into account for the next paper. So we've instituted that in all of our papers, not just for COVID-19. And I found it to be quite useful in just initiating even more productive dialogue between the authors and reviewers. So that's been one thing that we've done. And then I would say the other shift is that this digital environment has really allowed us to get closer to researchers and younger researchers in ways that I hadn't anticipated. So I would say one of the highlights of this past year for me was the trainees of the Till and McCullough meetings in Canada were organizing a workshop about science communication, and they invited me to come talk about what makes a good review article. And instead of just giving a talk, we realized that we had an opportunity to do something far more interactive, because we had all this extra planning, and it was really important to me to do something that actually engaged the trainees, understanding that it's just such a difficult time to be in a lab right now. So we kind of gave them a homework assignment, which was a review article that was under review. And they broke into groups and picked different parts. I assigned them different parts of the review article, and then they did a peer review of it. And then at the workshop, they presented on it, and I asked them to compile it all, and I gave it to the review authors, which in this case was Gordana Vuniak-Novak-Kovovic. And so you'll see in our current issue, the whole thing has been published. So we published the review, which took into account these trainees' really brilliant suggestions about how to improve the article. They were so thoughtful. And then the peer review itself. So I don't think that this would have happened if it hadn't been for the pandemic and for these kind of digital opportunities to meet with not just the heads of labs, but the trainees that are working with them. So those are some positive takeaways that I've had from this experience?
I agree 100%. I think there's a lot of elements that are going to stick around in the post-COVID era when we come back into each other's company. They'll still have a lot of the digital residue, although the scoring, the gamification of the conference needs to stop, because I'm just getting upset at my score.
He's so obsessed about this. He's looking at his score every single day. He's like, "Oh, what's your
My
score?
score
What's your score?"
doesn't make any sense. Okay, guys, I'm confused. Anyway, moving on. The reality, though, I think what you're alluding to there is, like, the quieter voices in the room, not just the big heads of labs, but also the trainees. And that brings me into my question. In this conference, we've talked a bit about that. There was equity in science panel, which was, I thought, great. And it's nice that year after year, I think they're trying to pay attention to these more social issues. And there's a women in science panel. This is a year where we're really talking about equity, right? And access and inclusion and, privilege, really. And I'm on 100. I happen to be mixed race, but look at me. I have some white privilege, right? I have some male privilege. And I think one of the first things of privilege is you got to just shut up and, kind of say, I have the privilege. I don't get to talk about the privilege, right? And when I think about science, it's interesting for me as a scientist who hasn't really quite gotten the big juice yet. As a scientist, I'm getting there. I'm working on it. But there's an element of science where scientists, they earn their privilege and then they flex their privilege forever, emeritus, right? You become a scientific star and you don't get dislodged, except in rare cases, usually involving controversy or kind of scandal or fraud, whatever. But, to put it short, the rich get richer. The more published get more publications, more grant funds and all that stuff. And your place, STEMCELL, you're in a position to shake that up a little bit. And I know you've done a lot already toward that end, but fundamentally, it seems really difficult to change the situation because of that one thing, that you earn the privilege. No one ever says, I earned my whiteness. There's a fundamental thing of privilege in the other context where it's not deserved, right? So you should divest from it. But how do you say you divest from something you earned, or should you? Do you think that the system, this is my question, do we have to have some kind of radical shakeup to change the review system, the tenure track, just the system of recognition in science? Or is it not much to be done? It's a fundamental, integral, intrinsic limitation.
Mm-hmm. No, I fully agree with you. And we've debated this quite a bit within our publication group. We obviously know that there are structural issues. There's structural racism. There's discrimination based on gender, sex. And what we've discussed is, what is our role as publishers in this process in science and in making science more equitable? Where can we actually make an impact? And so we've come up with some things, and it's been interesting to see the way that our company has actually responded to this. So the first thing is that we've hired an inclusion and diversity officer who's part of our senior management team. So this person, part of their role is just focused on this, and they have the eyes and ears of the people making the major decisions at Cell Press. And so her name is Cal Lim. And so she's been charged with really looking at, how we can promote diversity and inclusion in science. And so some of the actionable things that she's done so far, and I'm sure there's going to be more to come, is instituting things like an inclusion and diversity form that we now require every single author to fill out on their papers for publication. And so what this does is it reports not only on the science, but on the demographics of the authors. And so we have several blog posts about it, and our VP of editorial, Debbie Sweet, has spoken about this at several conferences if you're looking for more information on it. But basically, we ask, were there people of underrepresented minorities involved in this research? Was there funding for that? How did you think about sex and gender in terms of the subjects of your research, both for animal models, but human subject research, and really making sure that there's an opportunity for authors to make this more publicly available. It's one small step. I know there's plenty more that we need to do. We've also begun to look more carefully at the demographics of our customers and the people that we serve. And so now at STEMCELL, if you and many of our journals, if you submit a manuscript, we ask you to give us some demographic information. I think before there was kind of a hesitancy to ask authors about who they are, because there was thinking that you should be not paying attention to things like gender or race. But if we don't know who is reviewing our papers, who our authors are, then we can't really even look and see whether we have a problem and think about how to solve that. So now we are we get this information, and so at STEMCELL, it's definitely something that a part of our bigger strategy discussions we talk about. We look at our author pool and our reviewer pool. We look at whether or not the submitted authors reflects that are women, reflect our published authors, and in fact, women are very successful at STEMCELL. About the same rate that submits to the journal is published with the journal. So that helps us look at what our biases may be. We certainly have some work to do in terms of, I think, geography. Our authorship base is very Western centered, especially the US, and so that's one of the reasons why I've expanded our editorial team to exist both in Shanghai. We're about to hire someone in Europe. We're on the East Coast and West Coast of the US, so we have more access to Asia and Europe from there as well. I want to make sure that we take a real good look at what we're doing and try to make steps to rectify that. And then I would say the last thing we're doing in terms of thinking about inclusion and diversity is, we have the privilege, but it's not our voice that needs to be heard. It's the underrepresented minorities that need to have access to our platforms. And so you'll see for example, Cell has published quite a bit about funding Black scientists, what the NIH is doing, but then actually from scientists themselves. We started a new award this year called the Rising Black Scientist Award. And so it's a scholarship. One is given to a postdoc scholar that it's like an essay contest, and we have a selection committee that picks them. And so there's a postdoc and a graduate student that were selected. Their essays were published in Cell and they were fantastic. I recommend reading them if you have a chance. And the last thing I would say is at STEMCELL, we're really interested in early career researchers because we see that there's a more diverse pool. People coming from far different backgrounds, a wider range of countries. And so trying to make sure that we engage with scientists that are coming up now will help us be more thoughtful as we look to the future.
Wow, I didn't expect such a comprehensive effort, and I have to my hats off to you, because it's such a tough problem, and it's, I mean, I don't know about you, but it never occurs to me when I look at a new article, it never occurs to me the color of the skin of any of the authors, male, female, any of it. It's the science, and the truth of it. So like, it's not something that would jump out at me as like, it's the problem of the journal editors. I mean, obviously this is a systemic issue. And I think there's a lot of resources, well, not enough, but there's some resources being dedicated to addressing the problem at the root. And I mean, it's really, I think, very heartening to hear that you're not only tackling it at the top, but also doing, I think, your part to try and, tackle at the root as well. So I commend you and all the Cell Press group that's got of the same mind on this. And thank you so much for joining us for this Day 4 episode. Can't wait to see you next year in person in San Fran. We can have a drink, maybe, coffee, maybe something.
Yeah, I love that.
PG. But that brings us to the end of this episode for our listeners and watchers. Don't forget to follow us on Twitter at STEMCELL Podcast to find out what we're doing at the meeting, and check back here tomorrow for our next and final episode That's going to be with Nicolas Rivron. It's going to be an exciting bookend to a conference that's been very much centered on these human stem cell based embryo models. So that's going to be really exciting. We've talked about it at length on this and previous episodes of this video series. Thanks so much for watching, you guys. We'll see you hopefully tomorrow. Thanks, Sheila, for joining us.